Hey, hey, hey! Part 2, let's go. We're going to talk about proteins and nucleic acids. Alright, so proteins. This is what a protein kind of looks like. I like to think of it as a gigantic ball of yarn that a kitty cat played with and left laying on the floor. Just kind of looks like a bunch of squiggles. It also looks like a preschooler drawing. And then them going, "That's my best friend Joe." That big old pile of squiggles. So proteins. Know that we also know these as polypeptides or enzymes. Enzymes are just an example, a very particular example of proteins. Please remember, this is why I tell you, if you ever meet someone and you're like, man, I just think that person's just so nifty, you can tell them, you're a pretty bunch of proteins. Or you're a handsome bunch of proteins, because proteins, please highlight that it makes up greater than 50% of an organism in its dry weight. We also call that biomass. So remember, just like every other macromolecule, structure equals function. So we have to know the monomer, the building block. Those building blocks are amino acids. There are 20 different amino acids in making proteins. And then an individual amino acid, a monomer, is bonded together by a peptide bond. Please make sure you highlight that, because it's going to come back to play when we go into our 2 DNA units coming up. And please highlight that proteins and enzymes are the workhorse of the cell. They carry out all kinds of functions. If you're an enzyme, we're going to find out that those break things up or build things up. And that's just a type of protein. Please know that the arrangement and quantity of amino acids affect the structure and function of that protein or enzyme. Ooh. So, some amino acids. Think about those carbon groups. Can you spot an amine group? Can you spot a carboxyl group? Ooh. Amine, what element are we looking for? We're looking for that nitrogen. See, there's a nitrogen in there. Nitrogen, 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 nitrogen. Carboxyl. Well, if I'm a carboxyl, am I just double bonded to one oxygen? Or am I double bonded to one oxygen and a single bond to another oxygen? Oh wow, looky there. This time that single bonded oxygen isn't attached to an H out here. No, just got a single bonded oxygen. Amine and carboxyl, they came back, guys. So more common amino acids. These particular ones, if we look at the previous one, the ones that are listed here, your amino acids can be polar or nonpolar. Remember, nonpolar, if we look over here, means hydrophobic. These hate water. Because look at all these hydrogens in here. But if we head over to these are hydrophilic. Now, why in the world would these be hydrophilic? They still have, I don't understand. Why is that? Because they have charges. They have positive, negative charges. Because look right over here. That oxygen has a charge. Charge, 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 charge. All of these have charges on them. Ooh, there's a charge right there. You will not have to know that for the test. It's just a So peptide bond, remember peptide bonds hold individual amino acids together. And let's think about this. We talked about dehydration and we talked about hydrolysis. Remember, dehydration means that we are taking a hydrogen from a smaller molecule and an OH group, a hydroxyl, from another kind of a bigger molecule, and we're going to take that OH plus that plain old hydrogen and H, and we're going to bring those together and it form water. So let's look. It happens right here. Ooh. Look, there's an OH plus just a regular H. It's going to come out here and it's going to, those two H's are going to form a bond. They're going to link up with that oxygen and it's going to take out water. It's going to be dehydrated. So, what type of chemical reaction is occurring? We take that hydroxyl plus a hydrogen. It's going to remove his water. Dehydration. If this was hydrolysis, we would not be taking out water. We'd be adding in a water. But we can't do that right here. We can't. If we wanted to split up this peptide bond, we would have to go through hydrolysis or hydrolysis, however you want to. You would actually have to add a water. That water would split up and break that peptide bond. And it would actually go back to being this molecule. If we wanted to turn it back into this molecule, we would combine that hydroxyl and that hydrogen. It would come out as water, and then it would form this molecule again. So you're either adding water to break something up, or you're taking water out to build something bigger. Alright, so let's look at primary, secondary, tertiary, and quaternary structure. Primary structure, just go ahead and draw you this primary structure. I'm going to draw it with you. Primary structure means that it's the sequence of amino acids. So let's just draw one. It just kind of looks like this. It kind of looks like a wavy line. Just draw that out to the left there where you have some space. It looks just like that. So there's your amino acid. That's your amine end. Here's your carboxyl end. That means it's an amino acid. Please highlight that it's the sequence. And this one would actually be, an example would be insulin. If you think about insulin, that's usually the medication, the protein that people that have type 1 or type 2 diabetes, if they have very high blood sugar, they will actually take insulin to lower their blood sugar so that it doesn't reach too high of a level and cause damage inside the kidneys or the pancreas. So, out beside where it says little letter D, this is sickle cell disease, which we're going to, this is where the 6th amino acid was changed in the primary sequence. And instead of one amino acid, instead of it looking like these cute little donuts, it turned into these sickle-shaped cells. A sickle was actually a piece of farm equipment that people used in the Middle Ages up until they created better equipment like plows. It was actually a metal crescent-shaped piece of metal, and it was stuck and attached to a wooden handle. And people would actually cut their wheat in their crops by swinging this toward the ground. And that blade, that crescent-shaped blade, would cut the grass. So that's what they named it after, sickle cell disease. It is just because you changed that amino acid. Please remember that I don't make your outlines. Someone else does. So if you want to, and it's perfectly fine with me, you can mark out that phrase that says the easiest way to remember this is the 6th amino acid that changed. You can mark that out because, that's just not very pleasant. So, secondary structure, let's look at that. This talks about the hydrogen bonding between those neighboring amino acids. This allows your amino acids to start folding up. Ooh. We usually have, let's draw these out beside there. We usually have coils. Oh, doodle, I did it again. So let's draw these out there. We can have coils like this, or we can have structures like this. These are actually called alpha coils, and these are called beta pleats. You do not have to know those names yet. We'll talk about those later. So just draw you 2 examples of secondary structure and highlight that it's the hydrogen bonding that gives the protein some flexibility. You're flexible. So here's some pleating. Ooh. Here's some coils. Alpha coils and beta pleats. Tertiary structure. Please highlight that this is a variety of bonds. This could be covalent, ionic, hydrogen, between distant amino acids. These are not amino acids that are neighbors. This is an amino acid that's in the same neighborhood as each other, but they're like 3 houses down. And we're gonna kind of draw this a little bit. This is where you have an amino acid. Just draw yours like this, and then draw you some bonds in here. You're linking up distant amino acids. Label that the hydrogen bond. This is another bond. This is an ionic bond. Put that this is a disulfide bridge. That will be very important later. So just draw you this kind of S-shaped structure. Make you some bonds in between there, and highlight. I'll highlight them for you, and name what type of bonds those are. Remember, tertiary structure, trio, or terti meaning 3, means that we are connecting and making a deeper and more complex shape. We are connecting amino acids that live in the same neighborhood, but they're a long distance away. They're 3 or 4 houses separating the 2. And then we come to the end of the road. Quaternary. Phew! Quaternary meaning quan 4. Please highlight that this is 2 or more polypeptides that are woven together. And your example is going to be hemoglobin. So we're just going to draw one out there. Oop, that's a highlighter. No! I've still drawn with a highlighter. Oh my goodness. So we're just going to draw this number. This is what a quaternary structure looks like. It just looks like a big old ball of yarn. Just make you a semi. It kind of looks like a little bit of a blob, like over here. Draw you a blob. That's quaternary. So protein's natural form. This is what most molecular biologists are going to draw a protein to look like. That has your primary, secondary, tertiary, and quaternary structure all drawn in one. Dehydration of a protein. Dehydration is just a very fancy word for, we basically destroy the protein. We mess up the shape. This is a normal protein. See how nice and squiggly and ball of yarnish it looks like? What happens is denaturing what happens is you have something changing, like the pH, the salt level, the temp. Something happens. It either goes significantly higher or significantly lower, and because of that change, it basically makes your ball of yarn, your protein, unfold. If your protein unfolds, it cannot do its job anymore. Because remember, structure equals function. So you have denatured, or you pretty much unwound your protein. You broke it up. That's terrible. So please highlight that denaturing, enzymes as well, because enzymes are a type of protein. This is the unraveling of a protein or enzyme, causing it not to function. This is the correct shape of that protein. This is not. In this shape over here, it can do its job. But if we denature it, we unravel it, it cannot do its job. Poor sad little protein. Then please flip over to the very last page. Please highlight that denaturing can be caused, like we said, by pH change, salt, temperature, and the most common bonds. It says what bond do you think is going to be broken for this to happen? Think about your weakest bond that we talked about. We talked about 3 of them. Highlight that common bonds that have been affected during denaturization, that unraveling, are the weak hydrogen bonds and the secondary structure. So you break those weak little hydrogen bonds, your poor little protein's just going to denature and unravel. It's going to be sad. It can't do its job. Hydrogen, like we just said, they're weak. Nucleic acid, on to the last one. Woohoo! Please highlight that the monomers, the building blocks, are nucleotides. The polymers, the big guys, the macromolecules, those are DNA and RNA. We're going to actually draw one out here. This is basically a 5-carbon sugar. I like to say that the 5-carbon sugar, let's just draw this. 5-carbon sugar looks like a house. And what happens is, attached to this house, on one side, is a phosphate group. And then over to the side over here, you have a nitrogen base, an amine group. It's always off to one side. Some of these will actually be, the phosphate will be in a different location, and the nitrogen can be in a different location. So please remember, the DNA and RNA highlight that these are the source of genes and heredity information primarily. This is what tells you what you look like. If you got your mama's eyes and your daddy's nose. So there are 2 types. So know that DNA, it is the polymer. We call it the master $1 million blueprint. Because it is kept safe in your nucleus. It's your original copy. And then RNA, we call it the cheap 10-cent copy. We make RNA from DNA. So we say that it's a disposable, recyclable copy of DNA because it makes messengers and other molecules. And then we have 2 types of nucleic acids. We have pyrimidines and purines. So let's draw a pyrimidine and a purine. Let's erase our little nucleic acid real quick. So this is, right up here, these are pyrimidines. So let's draw us one. Let's go with cysteine. So please, out beside pyrimidine, draw cysteine and its cute little structure. The reason why we ask you to do this is to know that it has a different name. That means that it has a different structure. So please highlight that it has 1 carbon ring in the nitrogen base. Oh, wow. And then purine, let's go ahead and draw adenine out to the side there. This has a 2 carbon ring. 1, 2. This guy Just has one carbon ring. And then please highlight letter G. It is always a pyrimidine paired with a purine. In that example, write yourself out an example. Like C for cysteine, it always pairs with G, guanine. If you are thymine, this pyrimidine up here, you pair with adenine, a purine. And so on and so forth. So please highlight that the sequence determines what protein or enzyme you make. We're going to be making proteins in molecular biology. And remember, structure equals function. So complementing base pairings, remember T goes with A, G goes with C. Double helix structure. Please know that DNA, the two sides are complementary, meaning that they fit together. One side has information to make proteins and enzymes, and the other is just a protective cap to make sure that your DNA is safe. And please know under the genes and evolution, Roman numeral 4, just highlight that first one. The more nucleotide sequence, the genes that you have in common, the more closely related the organisms are. So let's think about it. We're all human beings. The only thing that separates you from the person sitting beside you in class, the percent of your DNA that differs from anyone sitting beside you in class is, put yourself this note, this is 1/10 of 1%. Makes you different from the other human beings that you're around every day. That's not even a whole percent, guys. That's 1/10 of 1%. Think about how small that is. So the next time that you think, you know what, we're just so different, just like, I'm not even like those people. Now, maturity you might be. Emotionally you might be. But genetically, we're not that much different at all. So I hope that was helpful, and I'll see you in class tomorrow.